Okay. Pag ganito, clear pa rin ang boses ko?
Yes po, Doc.
Yes po.
Sige. So, for this lesson, we will be discussing more on the antimicrobial resistance of the different important na organisms, and then we will also transition to some important aspects in bacteriology. So, bakit ba important ang antimicrobial resistance? Bakit ba natin siya tinatry pag-aralan? Why is it an issue? Because lately there have been instances of multiple drug-resistant organisms, and the drug resistance in bacteria have affected several clinical conditions, and nagkakaroon tayo ng difficulty in treating common organisms na agent ng, for example, UTI, pneumonia. So, the overall impact of antimicrobial resistance to public health has been severe for the last few years. So, if you can notice, meron increase of 8.4% to 92.9% of E. coli being resistant to ciprofloxacin for urinary tract infection. So aside from that, also yung increase ng Klebsiella pneumoniae from 4.1% to 79.4% na resistant to a quinolone antibiotic, which is ciprofloxacin, being treated for pneumonia. So anong ibig sabihin nun? If we will ignore the significance or the impact of this resistance na nag-a-arise, we will have difficulty of treating higher or severe forms of infection. So, ciprofloxacin, that is not a basic drug. Actually, we don't want to use that immediately if we think na merong UTI ang isang patient. Kasi sometimes, may mga other drugs na pwede naman na i-gamitin to treat UTI, or pwede naman na gamitin for Klebsiella pneumoniae. But because ciprofloxacin is highly effective as a treatment for infections, it has been utilized repeatedly. So the problem with that is nagkakaroon din ng rising na resistance. So lately, you will notice na ang daming nagpo-post ng mga health professionals kasi meron tayong antibiotic, si colistin, and we use it sometimes as the last resort treatment for life-threatening infections caused by carbapenem-resistant Enterobacteriaceae. So ngayon, if last resort na natin siya and it is not easily pwedeng bilhin sa market, tapos resistant pa yun na organism, so ano na lang ang next step natin for some other bacteria na mag-i-infect sa mga hospitalized patients, ganun. So the burden of resistance is increasing, and the scientists, or we as healthcare members of the team, has not been keeping up enough to overcome the widening resistance of organisms. So yun yung problem natin. Kasi hindi rin tayo basta-basta pwedeng magbigay or mag-shell out ng antimicrobial sa market kasi we have lots of phases or levels to consider. And hindi yun basta-basta natitreat kasi marami tayong mga pharmacologic things na we have to consider. So that is the burden of drug resistance. So aside from the general resistance, so yung Mycobacterium tuberculosis has been detected to have several multi-drug resistance against common antibiotics na ginagamit against it. And TB is very common in the Philippines. And if the common drugs that are being used or the first line drugs that are being used are ineffective, so we will have a larger burden in terms of treating the sick members of our community. So 3.4% of new TB cases and 18% of previously treated cases are now using last resort TB drugs. And that means a lot. And that was in 2018 pa. Hindi pa na-carry over yung pandemic period na halos lahat ng pwedeng ma-access na antibiotics ng other people is ginagamit nila without proper consultation because of the fear of getting the virus. So, there is a tendency na nag-increase ito in a higher rate compared to the pre-pandemic situation. So, aside from your TB, there are also arising drug resistance in viruses. So, most importantly, yung antiretroviral drugs, yung ating HIV, na increasing din in census for the past few years, nagkakaroon tayo ng observing resistance on the first line of treatment of HIV infection. So, in terms of viruses naman, specifically here in HIV or antiretroviral drugs, for example, one patient, di ba, lifetime treatment ito. So, that patient is being treated with first-line antiretroviral drug. Tapos, here comes another patient na being treated with a second-line antiretroviral drug. And when those two will meet, sometimes the resistant strains doon sa patient B can be passed sa patient A because of repeated high-risk behavior. So that is one of the reasons bakit nagkakaroon tayo ng increasing resistance of HIV against antiretroviral drugs. So aside sa viruses and common bacteria, meron din tayong increasing resistance in malarial parasites. So I think so familiar naman tayo sa first line, which is yung chloroquine. And nagkakaroon na tayo ng combination therapies against Plasmodium falciparum. And the problem is, with those combination therapies, nagkakaroon din ng resistance ang Plasmodium species. So, so far, we have been efficient in trying to control the malarial na parasite burden in our country because of preventive actions. However, if we will reach a point na hindi na masyadong effective yung mga anti-protozoal drugs against your Plasmodium species, we will have a difficulty in treating arising infections. That's why lately there are a lot of studies being dedicated in studying the science behind Plasmodium species being resistant to the common anti-protozoal drugs. And also, aside from malarial parasites or protozoan, syempre yung ating mga fungal organisms are also learning to be resistant against the common antifungal. And it's not common right now, but if it will reach the point of the same degree or same rate doon sa mga bacterial na resistance against common antibiotic drugs, this will be a bigger problem because most of the antifungal medicine we have are toxic to the body. So, konti lang yung effective na antifungals. And if magiging resistant pa talaga yung mga current existing fungal organisms against those common antifungal na accepted to be given to patients, you will have a burden and you will have a problem in terms of treating fungal infection. And fungal infections are being treated in a long-term method. Hindi siya pareho ng mga antibiotic or antivirus or even mga antiprotozoal na pwede mga 1 to 2 weeks. Yung fungal treatment is aabot ng 3 to 6 months. So that is a difficult resistance rate that we have to overcome. So your antimicrobial resistance is a complex problem and it requires united approach. So yung One Health approach na pinapatry na i-apply ng WHO and Department of Health, we have to connect with the different sectors of our community so that we can try to overcome antimicrobial resistance. So that is the burden of AMR. That's why we are trying to study and formulate mga new methods in overcoming antimicrobial resistance. So specifically, yung Mycobacterium tuberculosis na resistance are known na increased among Asian population. Specifically, yung mga Southeast Asian population. And we belong to that population. That's why if we will go and magpupunta tayo sa ibang bansa, for example, Middle East, UAE, or US, parang sa atin, common na yung possibility of having tuberculosis. Pero sa kanila, if you were found out to have tuberculosis, it's either i-isolate ka ng very long period of time, about 2 weeks, before induction of treatment or administration of treatment, or sometimes pinapabalik talaga sa country ang patient. So yan ang nangyayari because very rampant sa atin ang Mycobacterium tuberculosis na infection. And in this course or in this subject, what we are gonna try to understand is the science behind antimicrobial resistance. Kasi we are advanced micro para. And hindi tayo more on yung public health. So paano ba nagkakaroon ng development of antimicrobial resistance ang microbes? So first, ang purpose ng antimicrobial products is to kill or significantly slow the growth of disease-causing microbes. Sometimes, yung conditions present can drive some of the microbes to resist antimicrobial activity, and yung antimicrobial resistance activity acquired by that microbe can be passed on to next generation through different methods, pwedeng vertical and or horizontal transfer. So it will pass resistance genes to other microbes and it will increase the quantity of the resistant pathogens. So this is a timeline of some of the antibiotic resistance being identified. So yung antibiotic resistance, totally, is it a responsibility of us as stewards of antibiotic treatment? Of course not, because there is a genetic component in the natural selection of natural evolution of life na eventually what antibiotics are effective before, after certain levels of evolution, hindi na talaga siya magiging effective on that type of microorganism. Whether or not meron tayong maling intervention na nagawa. But the burden that we add is when we are not being wise in using antibiotics. So, na-magnify yung natural na resistance development of these organisms. So, during the 1940s, nagkaroon tayo ng development of, penicillin-resistant Staphylococcus. And yung antibiotic na na-introduce natin, penicillin has been identified only given to the market around 1943. So even if we were not able to introduce the antibiotic, there were already organisms naturally resistant to it. So this is the sample timeline of the antibiotic resistance na na-identify. Ito. Tapos dito naman yung antibiotics na being introduced. So if you can correlate, mas malaki or mas mataas yung rate talaga ng antibiotic resistance na nag-a-arise sa community. So what are the several mechanisms of antibiotic resistance? Meron tayong tinatawag na intrinsic resistance. Meron tayong tinatawag na acquired drug resistance, genetic change, and DNA transfer. So when we say intrinsic resistance, that means that the bacteria is able to have that resistance due to the evolution and the effect of changing their structure or components. So for example, an antibiotic that affects the cell wall building mechanism of a bacteria, for example, yung mga penicillin or mga beta-lactam drugs, automatically it will not be effective against those organisms na wala namang cell wall. Kaya there is a thing that we call as intrinsic resistance. Pag sinabi nating acquired resistance, this is the resistance that can be obtained by being able to resist a particular activity of an antimicrobial agent when it was previously susceptible. So that organism can acquire that resistance, pwedeng genetic mutation. So the bacteria or the bacterium will survive by getting nucleic acid material or DNA from a bacterium that is already previously resistant. So one example natin is Mycobacterium tuberculosis that is already resistant to rifamycin. So that is parang a derivative of rifampicin that we already use against Mycobacterium tuberculosis. So for genetic changes, so yun yung isa sa mga major things that we study, yung mga DNA ng mga organisms ng bacteria can change after the production of protein. So when that happens, bacterial components and receptors will be unrecognized by the antibiotic. So those bacteria na sharing the same environment can have mga intrinsic genetic determinants of resistance now mag-alter sa kanilang structure, and it will make them resistant to antibiotics. And meron din tayong tinatawag na specifically DNA transfer. So yung DNA transfer can be in the form of transformation, transduction, or conjugation. So those are things na pwedeng maging mechanisms of resistance of a bacteria. So pag sinabi nating transformation, sometimes the bacteria can encounter some naked fragments of DNA. So yung naked fragments of DNA will carry antimicrobial resistance genes. And when those fragments will be taken into the cell through transformation process, that DNA fragment will be incorporated into the host cell chromosome by recombination, and the resulting cell will be resistant against an antibiotic. Pag sinabi nating transduction, that is when yung tinatawag nating bacteriophage or bacteriophage, and they are multiplying in the cytoplasm of a bacteria or bacterium. So yung mga fragments of DNA from the chromosomes or from plasmids can be packaged in a viral coat and enter another host cell, meaning enter another bacteria. Kasi di ba ang mga bacteriophage or bacteriophage are parang viruses of bacteria. So when those fragments na kinuha nila doon sa initial na organism contains genes that are resistant to a certain antimicrobial agent, so napasa nila yun doon sa next host cell kung saan sila pumasok. So that is transduction. And conjugation naman is when a bacteria contains extra chromosomal genetic elements. Ano yung tawag natin doon? Yun yung plasmids. So those plasmids can carry genes for antimicrobial resistance. So pag merong dalawang host cell or bacteria that are closely adjacent to each other, pwede silang magpasahanan ng plasmid through a structure called as your pilus. So yung structure na yon, it will form a bridge between two bacteria, and then yung plasmid from the initial host or initial bacteria will be replicated and will be transferred to another cell. And ang result is an antimicrobial resistance created sa another organism. So that's what happens. For a while, ha? Hello, okay. So again, as a summary, pag sinabi nating DNA transfer, pag transformation, meaning naked DNA incorporation. Transduction is somehow through the process of phagocytosis or engulfment of the bacteriophage. And conjugation is through direct contact by a bridge, most of the time a pilus, transferring plasmid from one host cell to another. So those are mechanisms of antibiotic resistance. So meron tayong specific antimicrobial resistance mechanism for organisms. Meron tayong sinasabing signaling molecules as a mechanism of antibiotic resistance. So itong pag-arise ng signaling molecules as a mechanism when there are bacterial strains na nag-a-adapt sa certain environment before and or after antibiotic therapy. There are some cases as figured by studies before na merong mga molecules na ginagamit ng microorganisms as cell signaling agents. So, those signaling molecules will be utilized to prevent the effect of antimicrobials to that specific na organism. So, in a bacterial genome, yung information of antibiotics na na-acquire ng organisms and other signaling molecules, they are being contained in a certain genetic na location. And the information will be activated, and that is what we call as resistance mechanisms. And it will be passed from one species to another. So, pwede yun na na-develop nila yung information of acquiring these signaling molecules from parang mga endogenous antibiotics present in the environment. So aside from signaling molecules as a method of organisms in acquiring antibiotic resistance, meron ding reorganization of membrane and the permeability as a response to antibiotic pressure or the presence of an antibiotic. So ito yung isa sa mga common natin na alam na mechanism when it comes to antibiotic resistance. So yung different types of chemical structure of membranes of organisms. For example, lipopolysaccharide. So that is a common component of gram-negative na organisms. And the acquired resistance to these, the acquired resistance of these organisms against antibiotics can be due to decrease of the permeability of the cell membrane. And that is a major change in the cellular barrier composition of an organism. For example, yung mga gram-negative bacteria natin, di ba meron silang extra protection given by their outer membrane. That's why yung mga antibiotics active against gram-positives, hindi siya active against gram-negatives. Or some organism na specifically targeting gram-positive type of cell wall or cell membrane. So yung lipopolysaccharide composition of these gram-negative bacteria will increase somehow the asymmetry in the membrane architecture and the cross-binding between the lipopolysaccharides and some cations will now render this organism na less permeable to some hydrophilic agent. So because of that, yung importance of lipopolysaccharide is considered as a virulence factor because it is contributing to the resistance to antibiotics. So yan yung isa sa mga complex system responsible for the response of certain bacterial strains to their environment. So, ano yung nagko-control nung structure ng lipopolysaccharide of these organisms? Yung mga host-derived signals and gene expression important in maintaining the integrity of the lipopolysaccharide na component of our gram-negative organisms or gram-negative bacteria. So your porins are parang channels. So this is also important in regulation of the membrane permeability of organisms. They also function as parang membrane proteins. So yung porins together with efflux pumps are parang contributing to a joint action. So there will be parang presence of these membrane proteins, and they will form channels that will traverse both the inner and outer membrane. So yung entry of the different types of antibiotics, for example, beta-lactams or fluoroquinolones are regulated. And there are cases of beta-lactam resistant strains of E. coli na nagkakaroon ng deficiency in the expression of these membrane proteins. So meaning the porin and the efflux pumps are parang the ones that regulate the entrance of antibiotics inside the bacterial host. So kung konti lang sila or nagkakaroon ng parang alteration sa kanilang structure, hindi nagiging smooth ang entry ng mga antibiotics inside the bacterial host, and that will increase the resistance of that organism against those bacteria. So sometimes there are noted na clinical isolates with modifications in the structure of their porins and are already identified in many gram-negative strains. So meron yung study that noted na if konti lang ang porins or efflux or yung porins lang, konti lang yung porins ng certain organisms, mas resistant sila to certain types of antibiotics like quinolones and tetracyclines. So that is an important aspect of antibiotic resistance. Kaya ang iba, pinag-aaralan nila yung genetic expression of certain organisms as their study to check if meron bang map in this area na marami ng existing na antimicrobial resistance against an antibiotic na being commonly used. Now, your efflux pumps naman are somehow responsible not just for allowing entrance of antibiotics or other na mga substances towards the host cell, they are also responsible in extrusion or parang palalabasin nila yung mga toxic metabolites, whether endogenous or exogenous sa host cell. So, if ever nagkaroon ng entrance yung antibiotics sa host cell, if yung efflux pump ng certain organism is active and able to efficiently extrude or leave or ipalabas yung component outside of the cell, so nagkakaroon ng removal of antibiotics conferring that resistance to the given drug. Kasi pinapalabas niya sa host cell ng bacteria. So hindi siya nagta-take effect because of the presence of efflux pump. So sometimes, if there is an overexpression of efflux pump, nagkakaroon ng increased rate of acquisition of mutation because the drug is being removed out of the cell. And that efflux pump is a common mechanism of antibiotic resistance of gram-negative bacteria, also mga mycobacterium na mga organisms na multi-drug resistant na. So, yun yung minsan pinag-aaralan sa studies ng mga other medical scientists if yung bang mechanism of the multi-drug resistant na organisms is correlated with the efflux pumps as being overexpressed. So if gradual ba or prolonged bang exposure of the bacteria to increasing antibiotic concentrations is nagiging promoting factor in the overexpression of individual efflux pumps. Thus, consequentially, it will lead to increased resistance of those organisms against the actual antibiotics. So, yun yung isa sa mga points of possible study. So, ito, anong structure nga ito na common in antibiotics? So, this is your beta-lactam ring. So, yung beta-lactam ng mga drugs are commonly being used against a broad members of the bacterial community, pero mostly, yung beta-lactam drugs are effective against gram-positive bacteria. And yung problem natin is yung mga organisms minsan, they are already able to produce enzymes against the effectivity of these beta-lactam drugs. For example, meron na silang capability to release enzyme, tinatawag natin na beta-lactamase. So, di ba, familiar yung beta-lactamase. So, may mga drugs na if may beta-lactamase sila, hindi sila easily affected ng mga beta-lactam drugs. So, yung beta-lactamase, ano ang action niya? Itong nakalist dito, these are some of the mechanisms of resistance of organism against beta-lactam antibiotics. So, pwedeng nagkakaroon ng hydrolysis of antibiotic because of the beta-lactamase or because of the modification of penicillin-binding proteins or the cellular permeability is being changed. So this is an illustration presenting some of the mechanisms of action of antimicrobial resistance. So yung sinasabi natin na presence of beta-lactamase, they will inactivate pwedeng yung mga penicillins or mga cephalosporins. And some of them can also inactivate both classes of drug. So yung mga gram-positive bacteria will release or secrete their beta-lactamase, and these beta-lactamases will inactivate the beta-lactam drugs extracellularly. And somehow, these beta-lactamases of gram-positive bacteria is different from the beta-lactamases of gram-negative bacteria na naiiwan lang sa loob ng cell ang beta-lactamase ng gram-negative. So, yung pag-inactivate ng gram-negative sa beta-lactam drugs exist in the periplasmic space. And doon sa gram-positive, yung mechanism of beta-lactamase is in the surrounding environment of the organism. So, ano ang common location of the genes that encode these beta-lactamases? So, yung mga genes na nag-encode ng beta-lactamase can be present on the bacterial chromosome, plasmid, or transposable elements. Hindi ako makasulat sa presentation, kaya tinype ko na lang, ha? So, ito na mga bacterial genetics are important in the development of some organisms ng beta-lactamase. So, for example, yung beta-lactamase gene ni Staph aureus is located on a plasmid. Yung beta-lactamase ni Escherichia pneumoniae, so that will make it resistant against ampicillin and arginine that is present sa chromosome. And yung beta-lactamase genes naman ng other gram-negative and gram-positive organisms can be present sa plasmids and transposable elements. So those are the different na mga locations of genes that will code for beta-lactamase. So meron tayong classification of beta-lactamases also. So meron tayong tinatawag na class 1, group 1, group 2, group 3, and group 4. So the classification scheme of those beta-lactamases are proposed according to the hydrolytic spectrum, susceptibility to inhibitors, the gene location, and the gene sequence or the protein sequence. So meron tayong two major classification systems. Makasulat na. Meron tayong two major classification systems, yung tinatawag natin na Ambler system, and yung another naman is yung Bush classification of beta-lactamases. So, ang Ambler system is based on molecular structure. Molecular structure of your beta-lactamase. Tapos, yung ating Bush classification naman is based on the substrate of your beta-lactamases. So, yung different na classification schemes are somehow parang combined and allow them to create the different 4 groups according to substrates and inhibitor profile. So, meron tayong group 1, group 2, group 3, and group 4. So, yung ating group 1, these are cephalosporinases. So, ang kanyang ang kanyang substrate are mostly cephalosporins. Yung ating group 2 are both penicillinases, cephalosporinases, and carbapenemases. So these are the targets of the group 2 beta-lactamase, yung penicillin type, cephalosporin type, and carbapenem type. So, ang isa sa mga issues ng group 1 is that hindi siya masyadong na-inhibit ng clavulanic acid. So, ano pala yung clavulanic acid? Di ba, sabi natin, yung mga beta-lactam drugs, for example, ampicillin or amoxicillin can be inhibited by the release of beta-lactamase of an organism. Ngayon, nagdagdag tayo ng additional na component ng antibiotics to counteract the beta-lactamase present. So, ano yung mga common na compound na we add together with beta-lactam drugs? Example nun is clavulanic acid. That's why meron tayong amoxicillin and meron tayong co-amoxiclav lang. Amoxicillin lang and co-amoxiclav. So, yung co-amoxiclav meaning meron siyang clavulanic acid to counteract for those organisms na may beta-lactamase. And yung mga group 1 na type of beta-lactamase present on organism, hindi sila na-inhibit ng clavulanic acid. So kahit na meron siyang additional component is hindi siya effective in treating the infection. If may group 1 type of beta-lactamase ang organism. In group 2 naman, present siya sa mga yung penicillinases, cephalosporinases, and carbapenemases. So these are generally inhibited by clavulanic acid, sulbactam, and tazobactam as additional molecule. So this subgroup is formed doon sa mga organisms na may kanong tatlong klase ng components. And this subgroup also somehow defines the rate of hydrolysis of some organism by the group 2 na mga beta-lactamase. So example, yung mga oxacillin, ganun. Group 3 naman na type of beta-lactamase, ito yung tinatawag natin na metallo beta-lactamase. So they are able to hydrolyze penicillins, cephalosporins, and carbapenems. And they are inhibited by EDTA compounds, but not by those mga inhibitor structures like clavulanic acid, sulbactam, and tazobactam. And the last group naman is yung penicillinases na hindi rin inhibited properly by clavulanic acid. So again, yung group 1, yun lang sila yung cephalosporinases. Group 4, penicillinases. Yung group 2, yun yung may mga penicillinase, cephalosporinase, and carbapenemase, pero able to be inhibited by the components clavulanic acid, sulbactam, and tazobactam. Yung group 3, same types of beta-lactamase but not inhibited by the additional synthetic molecules na clavulanic acid, sulbactam, and tazobactam. So yun ang difference. So that is one of the mechanism of resistance of organisms against beta-lactam antibiotics. So now we go specifically to the mechanism of resistance of Mycobacterium tuberculosis. So si Mycobacterium tuberculosis, meron silang primary or transmitted drug resistance, meaning intrinsic na siya sa organism, and meron ding secondary or acquired drug resistance. So ano nga ba yung mga 4 common drugs being used against Mycobacterium tuberculosis? So meron tayong isoniazid, meron tayong rifampicin, pyrazinamide, and meron tayong ethambutol. So yun yung mga tinatawag nating RIPE or RIPEs, yung mga first line of drug therapy against Mycobacterium tuberculosis. So let's go first to the isoniazid. So your isoniazid is also known as isonicotinic acid hydrazide. So ang mechanism of action ng isoniazid is to diffuse or penetrate the host cell dito penetrate the host cell. And then, since your isoniazid is a prodrug, meaning para siyang pro-enzyme, kailangan niya ng activation, merong certain component ang Mycobacterium tuberculosis that will allow the activation of your isoniazid. So yung enzyme being coded by this gene, the CAT-G na gene, that enzyme is yung catalase peroxidase enzyme. It will now activate isoniazid in its active form. And yung active product ni isoniazid will interfere in the mycolic acid synthesis through a NADH-dependent na carrier protein encoded naman by INHA gene. So yung enzyme that will activate isoniazid to its active form encodes catalase peroxidase is the CAT-G gene. Ngayon, yung enzyme that is required for the inhibition of isoniazid ng mycolic acid synthesis is encoded by INHA gene or INHA gene. So if na proceed na niya ang parang reaction, redox reaction na triggered by the NADH system, it will now interfere with the elongation of the fatty acids. When that happens, nagkakaroon ng inhibition of cell wall synthesis, and it will now kill the bacilli. So yung resulting na isonicotinol radical, this will be the one to react to the part of the fatty acid na synthesis or elongation. So, nagkakaroon ng inhibition of several cellular pathways. So, walang synthesis of mycolic acid. So, walang proper cell wall synthesis. And if yan yung main mechanism of action of your isoniazid in a susceptible TB bacilli, so yung common na mechanism of resistance of TB against isoniazid is when there is a mutation in the CatG gene. So if meron siyang S315T mutation, it will lead to inefficient production of isoniazid NAD product. So nagkakaroon ng inhibition sa microbial action of isoniazid. So dito, not effective kasi may mutation dito. So nagkakaroon ng ineffective na antimicrobial activity C, INH. And aside from that, na CatG mutation, yung INHA mutation doon sa kanyang promoter region found in position 15, nagkakaroon ng overexpression of INHA, and that will be associated with resistant isolates against isoniazid, and that will be a result na hindi na effective ang isoniazid against some strains of Mycobacterium tuberculosis. So this is the most common na source of isoniazid resistance ng Mycobacterium tuberculosis pagka merong mutation sa CatG and INHA genes. So when they are present in combination, nagkakaroon ng 84% increase in global isoniazid resistance. So that is a problem because kasali siya sa first line of therapy against Mycobacterium tuberculosis. So ito naman is rifampin and other rifamycin na mechanism of resistance. Si rifampin, together with its mga kamag-anak, is a broad spectrum antibiotic, and it is the most widely used rifamycin type of drug to treat tuberculosis. And ang mechanism of action ng ating rifampin is to inhibit mRNA synthesis. So nagbabind siya sa DNA-dependent RNA polymerase ni Mycobacterium tuberculosis. And when that happens, it will allow yung subunit of RNA polymerase to become inactive. So nagkakaroon ng decreased activity of RNA polymerase si Mycobacterium tuberculosis. And that is encoded by the gene RPOB. Ito. Another antibiotic effect or mechanism of action of your rifampin is when it allows increased permeability of its cell wall. So nagkakaroon ng labas at pasok ng mga components ng organism. And another is its inactivity of the efflux pump. So hindi siya nakakaalis ng toxic metabolites. So yan yung mga mechanism of action ng rifampin. And the most important part of this mechanism of action is yung coded by RPOB, which allows inactivation of RNA polymerase. So, ano ngayon yung common mechanism of resistance ng ating Mycobacterium tuberculosis against rifampin? So, the rifampin resistance is mostly encoded in an RPOB gene mutation. So, si RPOB gene mutation will encode RNA polymerase. So, once pumasok ngayon si rifampin doon sa host cell, si Mycobacterium tuberculosis, tapos since may mutation yung RPOB, iba na yung change or configuration ngayon ni RNA polymerase. So, hindi na nakakabind si rifampin or rifampicin. So, when that happens, nagsistart na ang resistance of the Mycobacterium tuberculosis against rifampicin. Kasi wala nang binding site si rifampin kasi na-change ang configuration. So, nagkakaroon din ng decreased permeability and increased activity of efflux pump. And what is notable with rifampin or rifampicin is its resistance most often and noted na never siya nag-exist as rifampin resistance alone. Unlike si isoniazid na there are some cases na may isoniazid na resistance ang Mycobacterium tuberculosis alone. Pero ang rifampin, most of the time, hindi siya nag-exist as a lone resistance. Meron talaga siyang partner. Halos hindi ka makaka-isolate ng rifampin resistant na solo na Mycobacterium tuberculosis. That's why ginagamit siya as a surrogate marker for multi-drug resistant tuberculosis. If naka-exposure na kayo doon sa parang gene expert na mga public health offices, minsan, di ba meron silang gene expert tool na nilalagay doon yung sputum specimen to check for rifampin resistance. And mostly, ang tinatake note lang is rifampin resistance kasi the resistance to rifampin has noted to be coexisting with resistance against other drugs. Kaya ginamit na lang siya as surrogate marker for multi-drug resistant na tuberculosis. So that is rifampin. And the mutation of resistance against rifampin is through RPOB na gene. So si RPOB, siya yung nag-encode for beta subunit RNA polymerase. Next drug is yung pyrazinamide. So yung pyrazinamide is also a prodrug like your isoniazid. Pero si pyrazinamide is activated by another enzyme, which is yung pyrazinamidase, nicotinamidase, yung PZASE gene, enzyme, ito. And ano yung nag-encode sa kanya? PNCA gene. So si pyrazinamide, once activated, it will become pyrazinoic acid. Si pyrazinoic acid will be pumped out of the bacterial cell by a certain efflux mechanism. And now, it will go to the parang cell wall area ng Mycobacterium tuberculosis and it will inhibit fatty acid synthase 1. And that will now disrupt the membrane and function of the different enzymes dito sa cell wall synthesis. So, nagkakaroon ng increased acidity ang Mycobacterium tuberculosis. So, it somehow affects the MTB in that way. So, ang kanyang action is most active against non-growing tubercle bacilli than against young actively replicating organisms. Ang ginagawa kasi ni pyrazinoic acid, it will disrupt the bacterial membrane energetics and it will inhibit membrane transport. And the pyrazinamide will now destroy the integrity of the membrane of the organism, Mycobacterium tuberculosis. Kaya si pyrazinamide is more active against mga dormant na type of tuberculosis organism. Kasi hindi siya ganun ka-effective sa mga young actively dividing na mga Mycobacterium tuberculosis. Kasi may increased mechanism sila na hindi i-activate yung pyrazinoic acid and it will not be able to actively destroy the membrane. Ang effectivity ni pyrazinamide affects more on the dormant stage ni TB. Di ba ang ating tuberculosis will trigger a type 4 hypersensitivity reaction. And the hallmark of a type 4 hypersensitivity reaction is the presence of a granuloma. Tapos yung mga some organisms of TB there are somehow yung mga old non-growing tubercle bacilli, and the pyrazinamide itself is effective against those types of Mycobacterium tuberculosis na na-present sa body. So, ginoin siya with the first line of therapy because it helps in preventing relapse. Kasi yung mga dormant na mga TB organisms na naiiwan is naka-counteract ni pyrazinamide. So, saan pumapasok ang ating mechanism of resistance? So, doon pa rin sa mutation in the PNCA gene na nagko-code kay pyrazinamidase. So, if that gene is mutated, it will not activate pyrazinamide against to its active form, which is yung pyrazinoic acid. So, hindi rin mako-continue ni pyrazinamide ang kanyang action against Mycobacterium tuberculosis kasi wala yung enzyme that will make it an active drug, and it will remain as a prodrug in the organism and will not affect the tuberculosis na cell membrane. So the mutation is present in PNCA gene. So yung destabilizing membrane potential na activity ni pyrazinamide is not present if it is not transformed to its active form. So that is the mechanism of resistance ng pyrazinamide. Now, ethambutol. So, ethambutol is an antimycobacterial drug that has a poor sterilizing activity compared with the normal regimen isoniazid, rifampin, and pyrazinamide or streptomycin. Pero, because your ethambutol is not that toxic and it's able to affect the RNA metabolism and cellular pathway of Mycobacterium tuberculosis. Ginagamit siya as empiric treatment of individuals who are somehow treated as having increased risk of INH or isoniazid resistance while waiting for drug susceptibility results. So ang action ni ethambutol is to inhibit the arabinosyl transferase 3 enzyme, and that will destroy or cause disruption of the transport of the arabinose sugar, and nagkakaroon ng impairment of biosynthesis, so cell wall pa rin ang ina-attack. And the resistance of Mycobacterium tuberculosis against EMB is still a cause of certain point mutation in the EMB CAB operon. So that will involve amino acid substitution, and thus it will not allow EMB to factor in inhibiting the enzyme. So ang code natin dito na gene is yung EMB CAB operon. May EMBB and meron ding EMBA and meron ding EMBC. So those are genes that code for mutation of the tuberculosis, making it resistant against ethambutol. So yan ang mechanism of resistance. Meron tayong tinatawag, di ba, na Mycobacterium avium, and sometimes they have yung tinatawag na gene naturally present in them, and yung EMBA and EMB gene can be transferred through a plasmid vector and will be transferred to Mycobacterium smegmatis or Mycobacterium tuberculosis, making them resistant to EMB. So si Mycobacterium avium is inherently resistant to ethambutol, some strains. And yung gene niya is pwede niya i-transfer sa other Mycobacterium species like smegmatis and tuberculosis, and making them resistant also to ethambutol. So that is your ethambutol resistance. So those are the resistance of different drugs na Mycobacterium tuberculosis.